Translation is the process whereby the base sequence of a messenger RNA is converted into the amino acid sequence of a polypeptide. This process involves an mRNA, tRNAs, amino acids, a ribosome, and other accessory proteins. mRNA consists of three main parts. The portion from the 5' end to the translation start codon is the 5' untranslated leader region, more simply called the leader. The central portion is called the reading frame. It commences with the start codon, AUG, followed by a series of 3 nucleotide codons, each of which specifies an amino acid, and it ends with one of the stop codons, UAA, UAG, or UGA. The final portion of the mRNA consists of the section from the stop codon to the 3' end, called the 3' untranslated trailer sequence, more simply called the trailer. mRNAs vary in length because of differences in all 3 parts of this molecule. Transfer RNA functions to bring amino acids to the growing polypeptide during translation. It consists of a single RNA chain of about 75 to 90 nucleotides, each tRNA having a different sequence of bases. After it is transcribed, the tRNA folds up into a characteristic cloverleaf structure. The anticodon contains a 3 nucleotide sequence that is complementary to a codon on the mRNA. The amino acid is attached to the 3' free end at the top of the cloverleaf. The ribosome assembles amino acids into a polypeptide. Ribosomes consist of 2 parts, a large subunit and a small subunit. Ribosomes are made of ribosomal RNA and various proteins. The active ribosome contains 3 tRNA binding sites, namely E, exit, P, peptidyl, and A, aminoacyl. In prokaryotes, translation initiation occurs in 3 steps, as follows. First, the small ribosomal subunit, 30S subunit, complexes with 3 proteins named initiation factor 1, initiation factor 2, and initiation factor 3. A molecule of GTP also attaches. The ribosome IF-GTP complex recognizes a sequence in the leader, upstream of the AUG start codon. This sequence is called the ribosome binding site, or the Shine-Dalgarno sequence, after its discoverers. This sequence is complementary to a sequence at the 3' end of the 16S ribosomal RNA of the 30S ribosomal subunit, and serves to align the ribosome on the mRNA in its proper reading frame. Second, translation begins at the start codon AUG, a few bases downstream of the ribosome binding site. The first AUG in proteins specifies a modified form of methionine. This modification consists of the addition of a formyl group to methionine, making it formylmethionine. The anticodon of this initiator tRNA containing FMET complements AUG. Upon binding of FMET tRNA to mRNA, IF3 is released, with the remaining assembled components called the 30S initiation complex. Lastly, the large ribosomal subunit, 50S subunit, binds to the 30S initiation complex. GTP is hydrolyzed and IF1 and IF2 are released. The final complex is called the 70S initiation complex. The FMET tRNA is now situated in the P site of the ribosome, with its anticodon hydrogen bonded to the mRNA start codon. Initiation is now complete, and elongation. Once translation initiation is complete, elongation begins. The elongation phase consists of 3 steps: binding of a tRNA to the ribosome, formation of a peptide bond between adjacent amino acids, and movement of the ribosome to the next codon. Elongation continues 1 codon at a time until a stop codon is reached. As initiation concluded, FMET tRNA was hydrogen bonded to AUG in the P site of the ribosome. The next codon is in the A site. In this case, the codon is UCC, which encodes serine. The serine-bearing tRNA in the cytoplasm forms a complex with elongation factor Tu and GTP. This sernyl tRNA-EF Tu-GTP complex enters the A site where the tRNA anticodon forms hydrogen bonds with the UCC codon. Upon binding, the GTP is hydrolyzed and EFTU is released and recycled. With a tRNA in both the P and A sites, a peptide bond can form between adjacent amino acids. This reaction is catalyzed by peptidyl transferase residing in the large ribosomal subunit. The first step involves breaking the bond between FMET and its tRNA in the P site. Next, a peptide bond forms between the now freed FMET and the serine which is attached to its tRNA in the A site. Peptide bond formation is a condensation reaction that results in the release of water. At one time, it was thought that peptidyl transferase was an enzymatic protein. However, more recently, it was discovered that peptidyl transferase is a catalytic RNA molecule. An RNA capable of catalyzing a reaction is called a ribozyme. Once the peptide bond has formed, there remains an uncharged tRNA in the P site and a tRNA with a growing polypeptide chain in the A site. The ribosome now moves one codon along the mRNA with the help of EFG and another GTP. An EFG-GTP complex binds to the ribosome, hydrolysis of GTP occurs, and translocation begins. The uncharged tRNA moves from the P site into the E site, blocking the next aminoacyl tRNA from attaching to the A site until translocation is complete. The tRNA in the A site, still bound to its codon, now moves into the P site. Once this tRNA is bound correctly in the P site, the uncharged tRNA is ejected from the E site. The A site is now open for the next aminoacyl tRNA to enter, allowing the elongation process to continue. An average polypeptide consists of about 300 amino acids. Let's watch the elongation process once again. Elongation continues until the ribosome reaches one of three stop codons, UAG, UAA, and UGA. Unlike other codons, stop codons do not code for any amino acid, so no normal tRNAs have anticodons that complement these stop codons. The ribosome recognizes polypeptide chain termination with the help of proteins called release factors. RFs read the stop codon and initiate a series of specific termination events. E. coli has three RFs, RF1, RF2, and RF3. RF1 recognizes UAA and UAG, while RF2 recognizes UAA and UGA. RF3 does not recognize any stop codons, but stimulates the subsequent termination events. Elongation and ribosome translocation result in a stop codon entering the vacant A site. RF1 or RF2 binds to the stop codon, which triggers peptidyl transferase to cleave the now complete polypeptide from its tRNA located in the P site of the ribosome. The tRNA is then displaced from the P site of the ribosome, followed by disengagement of the RF and the 70S ribosome from the mRNA, and separation of the ribosome into individual 30S and 50S subunits.